Okay, so at this point, I would like to, we had a lot of great questions come in from our audience, and I want to make sure we address as many as possible. So first up, Loren, I'd like to address this question to you, and Cokey Nguyen, feel free to, provide your commentary too. Kelly asks us that in November 2023, the FDA announced several reported cases of lymphoma in patients who received CAR T-cell therapy products. What do you think about the potential risks of using T-cell therapy?
Yeah, again, I think on a personal basis, since I worked in the autologous space, my first instinct when that news came out was, is this being overblown to a certain extent, right? Are we talking about something that's at such a low level of risk compared to the benefits that, is this, a tempest in a teapot, if you will? But as we've gotten a little bit further into it and people start to see some linkages, I think one of the things that we've taken solace in, at least on the Poseida side of things, is that we've taken a non-viral approach, which we think is going to be one of the keys to having a far superior long-term safety profile. Again, you've seen the black box warnings that come out with it. I think that is more than sufficient in terms of making sure that people are aware of it. I think the good news is auto has shown what its benefits are. I think we've also recognized what the limitations are, and that's why we're working so hard in the allo field. And as we're getting there, we need to be aware of what the signals are. Again, when you're working on small patient populations, it's really hard to assess where you're going to be when you start talking about these rare occurrences. But I think one thing at a time, we start with the best design that we can put in place. And for us, that was a non-viral approach, which we think largely limits that. And I'm sure there's other companies that are working at ways to, again, you're talking about very small occurrence rates. How do you make sure that you've engineered the product to eliminate that to the best of your ability? You're never going to drive the risk levels to zero, but especially when you're talking about these patient populations that have such a high need, we have to figure out how to get those risks as close to zero as we can as we move forward.
Sure. Okay. Thank you. Cokey, I want to give you the opportunity to respond if you'd like to Kelly's question.
That's a great question. And obviously, everyone's been paying attention to it. And I think Loren and Poseida have an elegant solution with their integration process. We use a retroviral process. And so when we saw the guidance, it made us take pause and think about, obviously, how do you mitigate risk for the patient? And I think this is, again, where bifurcation happens between auto and allo. I think in auto, you get what you get, and you're under a time constraint because you have to manufacture, you have to do QC release, patients on bridging, you have to get that therapy into patients. There's probably, to be honest, less time to characterize if you're using lenti or retro, where is the virus going in those genomes? Obviously, the benefit to patients is far outweighs the rare risk, but you're going to get the drug. I think where allo, because you're doing batch manufacture, you've got probably more time to do QC. And what I mean by that is you potentially have more time to look at and characterize your lots, in the sense that you could look at, you do the sequencing and see where's the virus integrating, are they safe loci? You can do autonomous cell growth for longer, for example, 28 days. And if something's growing out, sure, you'll see a problem. And it's just, that just comes with the nature of allo. Excuse me. I don't think you have that time in auto. So it's a great question. Thank you.
Yeah, absolutely. Thank you to Kelly for submitting that question. I want to move on to Tania, and asks the question, what are your screening criteria for donor material? So Loren, I'd like for you to start off with a response to that question.
Yeah, it's a great question. And again, I think it's something that each organization will need to develop their own experience for, what your methodology is going to be for screening. But we have a three-step process. One is simply moving it back to our immuno-oncology team. They will take a sample and they will do a series of tests to just look at compatibility of the material with their expectations, just from a biomarker standpoint. If they are satisfied with the material, we have a small scale version of one of our processes that we will put the cells through, and that is strictly just to see how well they expand. So again, it's just a precursor of what we would expect in the manufacturing space. And then I think, as I mentioned earlier, the third step of our process is an an in vivo mouse model, where we assess tumor control on what the small scale final product would look like. So again, it's not apples to apples, just drop it into the manufacturing process, but that's kind of the point. You don't necessarily want to go through the full expense of putting it into a full scale process, go through all the components, all the media, all the labor associated with that. You want to keep it relatively modest in terms of how you go through the evaluation process. But it's certainly important to see. I'd be very reluctant to just take material, sight unseen, and move it all the way into the clinic and just hope, right, that it's going to work the way that you want it to. I think it's really important to come up with some screening criteria for your donors. And, when you start talking about what's the next level, do you start to figure out which ones perform superior, right, and go beyond just being adequate? Is there a subsection of the donors that are highly productive? And if that's the case, how do you find more of them, right? So there's always the next question. I think part of our job in CMC, part of our mantra is we create better problems to work on as time goes on. We're always going to have challenges. We just want challenges that are more about how do we get better yields, how do we get better quality, and get past the point of just getting through what I would call survival mode, from just making sure we've got enough donors and enough material. We want to think about what that next layer is and how do we thrive in this environment.
Sure. Okay, thank you. Cokey, I want to give you the opportunity to provide your thoughts too on the criteria for donor material for screening.
Yeah, I think it's one of the holy grails for allo is, what does a good donor look like? And I think a lot of us, and it could be platform specific, but I think one of the first principles is I think immune fitness, which is often related to age and BMI of a given donor. I think an added complexity for us at Atara is we don't do gene editing. So what we do is we do partial HLA matching. So that's our allo solution. So obviously we're looking at genotype, HLA genotype of the patient, the donor. And then we, like Loren alluded to, we do a small scale screening for alloreactivity. It's one of our important gatekeeping assays. We really want to make sure that as the donor T cells are run through our process, where we're minimizing potential donors, and you do see sometimes that have egregious, alloreactivity. And then there's, to Loren's point, there's a feasibility. How well does the T cell from this donor, this EBV specific T cell, how well does it transduce with the retrovirus? And then how well does it perform in our serial stem assay, which is kind of our surrogate, really, so that we don't go all the way into in vivo. So it's from the outset, it's eligibility, it's the HLA, and then obviously we have a certain criteria for what we think immune fitness looks like that goes into our donor selection.
Okay. Thank you, Cokey. I appreciate that. We're going to move on to another question. What are 1 to 2 lessons learned during the comparability exercise as you move from early phase to pivotal and commercialization? Either one of you can start. Cokey, if you'd like to, that's great. Or Loren, up to you.
Maybe I can go first. I don't have a story about pivotal or commercialization, but what I can talk about is some tech transfers that we did. We were actually internalizing something back from a contract manufacturer, and we wanted to make some improvements along the way. One of the things that we found to be challenging is, I think everyone always looks at a split donor comparability as the gold standard, and it is. But there are certain circumstances, if a certain unit operation has been problematic, right? The case in point that we've tried to make, and it resonated with the agency, was we don't necessarily want to show comparability to something that's been problematic or has been inconsistent or a struggle, right? So sometimes it's better to compartmentalize it and really just focus on that unit operation, demonstrate that there's differences between that unit operation, and hopefully you're superior with the change that you're going to make. Put that into place and then do a split run confirmation, or perhaps just pick your destination and do everything in triplicate, similar to what you did with your original IND. So there's a couple of different designs you can use for comparability, and I wouldn't fall into the trap of saying every single circumstance calls for a split run comparability, because that may or may not be the best tool to demonstrate that you're moving to a process that's stable.
Great. Thank you. Sorry about that, and my mute button again. Okay, I think we have time for probably one more question. And Cokey, this person has asked this specifically of you. Does the vector need to be purified for phase 1 trials?
You know, I think there's no strict guidance for phase 1. I don't think that you need to have a purified vector, but what I would say is, again, planning for pivotal, it's probably a good idea. What we talk about is phase appropriate quality and GMP. It's to really have that plan. So it's a lot easier, for example, if you start with purified vector. And what I mean by that is it makes your transition from your phase 1 process and phase 1 trial into your pivotal much easier. If you do a process change, and Loren's alluded to this, the recent guidance, it's an IND amendment, and it's basically a new clinical trial. So it's just something to keep in mind as you go into first-in-human studies. But yeah, it's probably a question we all struggle with: speed versus future speed.
Sure. And then, do you consider partial HLA matching for donor and patient?
We sure do. Yeah, it's one of the cornerstones for our EBV CAR-T platform is partial HLA matching. And you know, what we've seen in over 600 patients is we haven't seen graft versus host. We haven't seen any signs of host versus graft. So that we're just going with a biological approach for now, which hopefully complements what we're seeing with our gene editing colleagues.